Good morning, dear students. Let's begin our science class. Today I am going to explain you our biology chapter number 2, microorganisms, friend and foe. In our first chapter of biology, we discussed about crop production and management, and now we will be discussing about the microorganism friend and foe, where we will discuss about a lot of microorganisms and which microorganisms are useful, which are harmful for us, what are actually microorganisms, what are the types of microorganisms, the examples of microorganisms, and many more. So let's start. So our first topic for this will be microorganisms definition and its types. So what is a microorganism? An organism which cannot be seen by naked eye is called microorganism or microbe, and we need a microscope to see a microbe. Now that is one important thing, that we need a microscope for seeing any microorganism because it is so small in size it can never be visible from our naked eyes. There are 4 types of microorganism: bacteria, algae, fungi, and the fourth one is the protozoa. Now let's discuss about bacteria. Bacteria are microscopic organisms which have cell wall but do not have nucleus. Some bacteria are autotroph. Autotroph means they prepare their own food by using sunlight, chlorophyll, etc. But most are heterotroph. Heterotroph means that they depended on other organisms for obtaining their food. Bacteria are found in different shape like rod shaped, spiral, spherical, comma shaped. So rod shape is a linear structure, spiral shape is a different type of structure where we see it is a small rod like many structures are present. Then we see a spherical is round shaped, and comma shaped is just like a comma. Examples of bacteria you need to learn are Rhizobium, E. coli. E. coli means Escherichia coli, Pseudomonas, and Streptococcus. Streptococcus full name is Streptococcus pneumoniae. Now let's discuss about algae. Algae are green plants which are simple in structure. We have seen many times algae growing in our house also, wherever there is moisture and humidity. A cell of algae contains cell wall and chloroplast. Now we do not know what do we mean by cell wall and chloroplast because we have not studied what are they. But we can just see that chloroplast is something that gives green color to that plant. So here algae has a chloroplast, and that's why it has a green color. These are autotrophs. As they are having chloroplast, that means they are having chlorophyll. Means chlorophyll is consists of many small pigments called chloroplasts. So they are having chlorophyll, they prepare their own food. Some algae are microscopic. Examples are Chlamydomonas, Pyrogyra, etc. Fungi. We do not pronounce it as fungi. We pronounce it as fungi. Fungi have cell wall and nucleus in their cells but do not have chloroplast. So that means they are not green in color. They are saprotrophs. That means they grow on dead and decaying matter, and then they obtain nutrition from that. That is, they feed on dead remains of plants and animals. Almost all fungi are microscopic except mushrooms. Mushrooms we can see in some of the countries mushrooms are also eaten. Example of fungi are yeast, rhizopus, aspergillus, penicillium, etc. Let's discuss about protozoa. Protozoa do not have cell wall and chloroplast, but they do have a nucleus. Amoeba and plasmodium are the examples of protozoa. You have learned what do you mean by amoeba. You have seen it in your previous class also. So amoeba is irregular body which is actually a protozoa. In the previous class you only learned that it is a microorganism, but it's actually protozoa, and it is having a nucleus that we also saw in the previous class. Now the next heading for this chapter will be virus and habitat of the microorganisms. Now virus is a very important heading. It is a 5th category among the microorganisms. We do not classify virus in any of the classification system. It is just unique from anyone else. These are actually non-living when they are present in outer cell, but when they are present inside the cell, they work or start functioning like a living organism, and then they produce their own copies, and these viruses actually damage the whole cell. They control the cell, and then eventually the cell die, and in this way they cause the infection, and that's why these are considered as harmful. Viruses are also microscopic, but they are not considered as living beings. They are considered as a borderline case between living and non-living. A virus behaves as non-living when it is outside a host cell. That is, a virus does not carry on nutrition, respiration, reproduction when it is outside a host. So we say that whenever it does not get any specific cell on which it has to attack, it will remain as non-living thing. It is just seen like a machine, and it's actually very small in size than other organisms also. Means we discussed about algae, fungi, and we also discussed about bacteria, protozoa. Out of them, virus is more smaller than them. But once it is inside a host, it behaves like a living being. It carries on nutrition, respiration, reproduction when it is inside a host. An example of viruses are HIV virus. HIV full form is human immunodeficiency virus, tobacco mosaic virus, that is TMV, etc. Now, habitat of microorganisms means where the microorganisms are found. Microorganisms live almost everywhere on the earth. They are found on land, water, and in air. So, the mobile phone or the laptop or tablet on which you are seeing this, if you see it through a microscope, you find millions of microorganisms on your device. They are even found inside and on our body. It is also said that the number of cells that are present in our body are less than number of microorganisms, especially bacteria in our body. So in another way we can say that bacteria are constituting our body, not the cells. Some microbes are even found in extremely harsh conditions like in crater of volcano and in sulfur springs. Those are called thermoacidophiles and many other. Means they are living in the harsh conditions and hence their names. Now we will discuss about the friendly microorganisms. Means some microorganisms are those which are friendly to us, which are friendly in behavior and which help us a lot. Like lactobacillus is an example of bacteria. Lactobacillus facilitates the conversion of milk into curd. So the curd which we eat is due to the action of lactobacillus on it, and that's why the milk gets coagulated to curd. Yeast cause fermentation of many food items. The process by which sugar solution changes into alcohol due to anaerobic respiration by microbe is called fermentation. So yeast is also used and is also used as a batter of cake, idli or pakora. There is a production of carbon dioxide due to respiration by yeast, and carbon dioxide gas create bubble in dough or batter which makes fluffy cakes, breads, idli and dosa. So actually we say that yeast only makes our cakes fluffy. Fermentation also helps in production of alcohol from fruit juice, sugarcane juice or from cereals, and this process is utilized to make wine, beer and other alcoholic products. The next heading for this chapter is antibiotics. So what do we mean by antibiotic? It is a substance which kills or stops the growth of bacteria is called antibiotic. Means those bacteria which are harmful for our body, especially antibiotic acts on them and they kills them or they stops their growth. So that's why whenever we have a bacterial infection, we generally take antibiotics. Alexander Fleming discovered the antibiotic penicillin in 1929 during one of its experiments. Now this is one more question, that who discovered first antibiotic and what was the name of that antibiotic? So Alexander Fleming discovered penicillin. During one of his experiments, Alexander Fleming observed that bacteria did not grow on a culture where fungus or Penicillium notatum was present. So this penicillin which we see is actually a fungus, and here this fungus is actually when he was performing an experiment on fungus, and when on one of his plate or dish this fungus was growing, the bacteria was unable to grow over there. And why it was? Because it was that the Penicillium notatum, that fungus, was producing actually a chemical which was called as penicillin, and because of that chemical bacteria were killed or they were unable to grow on the fungus, and that's why we say that this antibiotic released by Penicillium notatum is called penicillin. Thus penicillin was prepared from a fungus Penicillium. Penicillin made it possible to treat many difficult diseases. Most of the modern antibiotics are derivatives of penicillin. Means the penicillin, that chemical which is obtained from Penicillium notatum, is now used to kill many of the bacterial cells and is used popularly as antibiotic in many other diseases which are bacterial related. Some examples of bacterial diseases are many, like there are provided, but here we are discussing just that medicines which are obtained from the antibiotic penicillin: amoxicillin, cefotaxime, cefoperazone, cefuroxime, ceftazidime, etc. Some other examples of antibiotics are tetracycline, norfloxacin, then ciprofloxacin, or ciprofloxacin, then doxycycline, etc. So these all are the antibiotics which we use for bacterial disease treatment. Now let's discuss the next heading of this chapter, that is vaccine. So what is a vaccine? Our body has the ability to fight disease. The defense mechanism of our body produces antibiotics to fight a disease causing microbe which may enter our body. While doing so, our body remembers to fight any future onslaught of that microbe. This concept was utilized to make vaccine against the various diseases. So it is the one of the important thing that whenever our body gets infected from any disease, the next time when the disease will attack on our body, the body will have a memory of the disease, and for the next time the disease effect will be less on our body. For example, if we are getting chicken pox, so it is a type of disease. So when our body goes to it first time, then we suffer from chicken pox, but then afterwards in our whole life we'll never suffer from this disease. It is so because the body has prepared such antibodies against that disease, so that whenever next time it will attack to our body will automatically kill it. So vaccine was such thing that when we observe this thing in our human body, so vaccine is actually a dose of those dead infectious microbes, those microbes which are causing the disease. Their dead form has been injected in us, and then afterwards, since it is dead, so it will be weak, so our body will then also prepare antibodies against it, and it will fight against it, and we will get cure. And now the next time when again the same disease in reality will attack on us, then we will not suffer. So that's actually vaccine. So vaccine is not a kind of medicine. Doesn't it surprise you that it is not a medicine? It is just that disease which you will be having. The dose of that disease or those microbes are given to you. A vaccine made by a weak or killed strain of disease causing microbe. When a vaccine is inoculated in the body, means injected in the body prepare antibodies against it. Thus the body learns and remembers how to fight with that microbe in future, and vaccination thus helps in preventing the disease against a particular disease. Now Edward Jenner discovered smallpox vaccine in 1798. So what was the name? It was Edward Jenner, and against smallpox. He observed that people who were rearing cows did not suffer from smallpox. That observation led him to discovery of smallpox vaccine. Vaccines are now available for many diseases like smallpox, tuberculosis, polio, tetanus, diphtheria, whooping cough, hepatitis, and many other. So here actually we are giving the same dead microbes or the poor microbes which are not having much power to the body. That's all for the day, students. Thank you and have a nice day.